Now look at this.
What's up, baby bio penguins? Today we do topic 7.10 on speciation. So speciation is when we get new species that come about. So what's the species? There's a couple different definitions for species, but we're going to go with the biological species concept, which states that two organisms are from the same species if they can interbreed and produce fertile, viable offspring. So there's two different types of speciations that we talk about. There's sympatric speciation, allopatric speciation. Sympatric speciation is going to take place in the same place, versus allopatric speciation takes place when they're apart. So traditionally, we're going to find that there is a geographical barrier with allopatric speciation that doesn't take place in sympatric. One of the leading causes of speciation has to do with reproductive isolation. There's a bunch of different types of reproductive isolation, but we're going to kind of focus in on the prezygotic and the postzygotic barriers. Prezygotic barriers are going to take place before the zygote forms. So this is going to be things that inhibit the interbreeding. There's 5 different examples. The first example we're going to talk about is temporal isolation. With temporal isolation, the two species are going to breed at different times, whether it's different times of the day, different times of the month, or different years. Habitat isolation, the two species are in different habitats, and due to these different habitats, they don't ever meet to mate. My favorite example, the blue-footed booby, the two are going to have different behaviors, or they'll have different mating rituals, whether it's a song or a dance, and due to those different rituals, they don't mate. Mechanical isolation has to do with that the two are unable to mate due to anatomical differences. And last but not least, gametic isolation. With gametic isolation, the gametes are unable to fuse, and if the sperm can't fuse with the egg, no zygote. With postzygotic barriers, this means that the zygote has formed and we're trying to inhibit future hybrids from forming. So our first example, reduce hybrid viability. The hybrid's not viable. It's sick, it's feeble, and it's not going to survive, so it won't continue reproducing. Our second example we have is reduce hybrid fertility. This means the hybrid is sterile, so it can't reproduce. So let's look at an example. The horse has 64 chromosomes, the donkey has 62 chromosomes. So the hybrid, the mule, has 63. Due to the odd number of chromosomes, it's unable to make gametes. Our last example is hybrid breakdown. First generation is perfectly fine. It is fertile and it's going to be viable, but all of the progeny that come from it have a reduction in their fitness. 3 things can take place when 2 different species meet in the hybrid zone. Reinforcement is going to reinforce reproductive barriers to inhibit the production of the hybrid. Stability is going to inhibit the gene flow between the parent species, so you end up having 3 different species: parent species A, hybrid species, and parent species B. And fusion is going to increase the gene flow and decrease reproductive barriers so the 2 species fuse into 1 species. AP Bio Penguins, just success. Bye, y'all.